Hello, everybody. I'm Katie Clapp, president and co-founder of FRAXA Research Foundation. And I'm really thrilled to be talking with Dr. Elizabeth Jonas today about her latest work on Fragile X syndrome. Dr. Jonas is professor of Internal Medicine and Neurology at Yale School of Medicine, and she's board certified in both fields. Her latest work was highlighted in an article in Yale News, which has gotten a lot of interest from the Fragile X community, and I'm sure others as well. And it's based on work published that she and her team published in the September 3rd edition of the journal Cell. And we will link both of those articles in the description of this video, so you can find them there. So, Liz, what can you tell me about this work on leaky membranes in the Fragile X mice? What's the significance and how did you come across these leaky membranes?
That's actually quite an interesting story. So we had been studying for many years mitochondria inside synapses, and our interest was originally in calcium release from mitochondria and how calcium affects synaptic transmission. But we quickly realized that mitochondria are actually plastic and they change dramatically during synaptic plasticity, so that they are becoming sometimes stronger and sometimes weaker depending on the type of synaptic stimulation that they are undergoing. And so we were fascinated by that, and we studied a protein called BCL-XL, which many people may know as a cell death protein, but we found that it was really important for synaptic strengthening. And so we were at a neuroscience meeting presenting our work on BCL-XL when we ran across a poster by an investigator at Columbia named Leaky. And he was also studying BCL-XL, but he was studying its role in the Fragile X scenario. And actually he was looking at the way BCL-XL affected the number of neurons in Fragile X animals. And he found that BCL-XL actually made too many cells, too many neurons in the hippocampus. And so we were really excited because we had already been thinking that BCL-XL could be playing a role in Fragile X synapses to help strengthen them or help them grow or mature. And so we started collaborating with Rick. And at that time, we became fascinated by some findings that actually, if you inhibited BCL-XL, the hallmark feature of protein synthesis increase that you would normally see in Fragile X was seen in wild type neurons. So instead of, we thought maybe BCL-XL could be downstream of Fragile X in some way, either hurting or protecting, causing too many cells or too many synapses. But instead of doing that, it was actually the opposite, that BCL-XL was trying to help. And that inhibition of BCL-XL was enhancing the already aberrant protein synthesis rate of the neurons. So we found that to be very exciting. So it wasn't just FMRP that could regulate protein synthesis rates, it was BCL-XL. And if BCL-XL could regulate protein synthesis rates, then that meant that there was something downstream of both FMRP and BCL-XL that was doing the regulation of the aberrant protein synthesis and growth of the neurons. And so, of course, knowing that BCL-XL affected mitochondria, we looked to the mitochondria to see if that was the cause of the aberrant protein synthesis rate and aberrant growth of the Fragile X neurons. And that's what we indeed found.
So the mitochondria, now just for those out there who don't remember their biology, where does the mitochondria fit in neurons?
So mitochondria, of course, are making ATP for neurons, and they're really important for synthesizing proteins and lipids. People tend to forget about that function of mitochondria. So the TCA cycle is really important for the synthesis of amino acids that you are not the ones that you don't eat, the ones that you make, and also for synthesis of lipids. They are in the neuron, in the neuronal cell body, but they're also out in the neuronal processes. So they're in the axon and they're in the dendrites, and they are populating the areas where synapses are growing. Particularly in larger synapses, you'll very often see a mitochondrion parked very close to the synapse, either amongst the synaptic vesicles or at the base of the dendritic spine on the postsynaptic side.
I believe that we first met you back when you were looking at BCSXL and funded a study that you did with Dr. Leaky.
Yeah.
So this current work kind of came out of that
Right.
early study, which was back in 2014.
Right, exactly. So FRAXA funded us when we thought, okay, maybe BCLXL is the bad guy. Maybe it's actually causing, it's upregulated in Fragile X, as are so many proteins, and that actually it's causing aberrant synapse formation. But it turned out that it was the opposite.
Yeah, isn't that funny? You go in with one idea and you think you've got the culprit, and it turns out to be just the other way around. But in a way, this is more exciting and
Exactly.
it sounds more fundamental. A leaky membrane sounds quite, striking, and it looked also from your work that these leaky membranes may possibly be involved in other diseases as well as Fragile X. Alzheimer's was mentioned.
That's correct. We haven't studied Alzheimer's directly, but we are studying Parkinson's disease. And we're very interested in a protein called DJ1 that's implicated in familial Parkinson's disease, and our studies indicate that without DJ1, you cannot close the inner membrane leak. And so patients who have mutant DJ1 can't close their leak, they can't make ATP properly, and their neurons also have reduced growth. And we published that in 2019.
So what are you looking to do next to follow up on this discovery?
Right. Well, a couple of very exciting things. So as you probably noticed in the paper, we're really interested in a drug, dexpramipexole, that we know can close the leak of the inner membrane of mitochondria. And the way it does this is by binding to the enzymatic portion of the ATP synthase. And you wouldn't imagine that the ATP synthase itself could have a leak in it. But indeed, this is actually the case, and we have located we know the location of the leak. It's inside the membrane portion of the ATP synthase in the center of the C subunit ring. Our work was at first controversial, but now is gaining wide acceptance. And the idea that this ATP synthase could be regulating itself by leaking. In other words, making it so that it can't make ATP efficiently at times, and then trying to, being able to regulate that leak so that it makes ATP more efficiently is really fascinating. And so there are a number of areas of interest that have developed out of this idea and out of the Fragile X findings. And one is, does this occur normally during development? So we were studying the Fragile X model, but now we're fascinated that during normal neuronal development, there may be leak closure, so that you are having a leaky inner membrane early in development, and that the leak has to close for proper synaptic and possibly neuronal maturation during brain development. And if it doesn't close at the right time, which is what we think is going on in Fragile X, then development will not take place in a timely fashion. So that's one really interesting area that we're looking at. And then, as you mentioned, in diseases, so we're extremely interested in neurodegenerative disease. We're focusing on Parkinson's, but there are other diseases of the brain, and there are diseases of the heart, of the kidneys, liver, diabetes, you name it. This leak is important for all of those diseases. And so we have many projects. What we found in the Fragile X scenario was really also another fascinating thing. We had anticipated this from earlier studies in which we saw that the enzyme could dissociate from the membrane, and that would leave the C subunit with the leak in it naked, so that the ATP synthase the enzyme wasn't there to close the leak. So that meant that the enzyme, which we also call the F1, is an inhibitor of the leak. In Fragile X model, as well as in other models, we find that the stoichiometry of the ATP synthase is abnormal. So there's not enough enzyme, and there's too much free C subunit, which is leaking. So therefore, you need proper protein synthesis to make the F1 or enzymatic component of the ATP synthase to close the leak. Now, do you do that normally during development? Do you lose the F1 in aging or in degenerative disease? We know that to be true in rodents. Is it true in mammals? Most likely it is. And so how can you prevent that? And that also raises the last project that I'm going to tell you about, which is that to repair Fragile X, not only do you need a drug like Dex that binds to the F1, but you need a drug that binds to the free C, because free C is leaking, and a drug that binds to F1 is not going to be able to repair that leak.
So this Dex is not an available drug, is that correct?
It's not. It was in trials for ALS. It passed phase 2 with flying colors, and that publication came out in Nature Medicine. But then in phase 3, in which all comers, 3,000 patients with ALS were tried, it was found to be ineffective. And of course, now subgroups are being analyzed. So does that mean that this mechanism that we have found in the lab, the function of DEX in the lab, does that mean that that's not working in ALS? Or does it mean that they have too much free C?
Right.
And
or
so
could the drug be also working on other pathways as well and kind of muddying the waters?
Maybe, although I don't think that we haven't found any negative effects of DEX in terms of side effects that are worth discussing here. There are certain immune effects that may actually be good. For example, in COVID, could be good, right? So we're working on that. But right now the drug is being tried. There's a trial going on in asthma patients. So they also work in immune cells. But as far as off-target effects that cause the lack of function during the trial, I don't think so. I think that it's a low affinity drug. It's really, really safe, and therefore may not be as potent as what you may want for treating this disorder. But also, you don't necessarily want a really potent drug. You want a drug that is allowing the ATP synthase to open the leak at times when it needs to, but then helping it close it during the maturational stage when you need it to be closed. So you don't want a high affinity drug that's going to slam it shut. But also, as I mentioned, we may be missing the boat if we don't think about the free C, because DEX will not work on that target.
Are there drugs that would work on free C?
Well, we're looking.
So that's one of the prongs going forward of your work.
Exactly.
I'm sure they're out there somewhere.
I think so. And, Yale has a good library. The NIH has a library. So we're studying that.
A lot of drugs on the shelf waiting for a good purpose.
For a home, yes.
For a home, waiting for a home. Well, thank you so much for talking with us. Is there anything else you would like to add about this work?
Well, I just obviously want to thank all the people who were involved. And I wanted to mention, Pavel Lishnersky, who's the lead author on the paper, or the first author, he put in so many years of work. And of course, it was an uphill battle because, FRAXA, of course, began to lose faith in us, and Simon's Foundation began to lose faith in us, and eventually we prevailed, which is a really exciting part of the story. But I also wanted to highlight, Rick Levy's work, and his paper also came out around the same time as our paper. He actually analyzed intensively the effects of coenzyme Q in Fragile X. And so that's another modulator, we think, of the leak that actually could be useful. And that's a really interesting paper. And also, I wanted to mention that so many undergraduates participated in this study. And we really had fun. We worked them hard and they worked hard. So they did mitochondrial ion channel recordings. They did they participated in the behavioral studies. They did Western blots. They were involved in every aspect of it. And for me, that was just really fun. So they're all on the paper with us, which is great.
I'm sure they're thrilled to be part of something so exciting. Well, as with all research, it takes a lot of money, time, people.
And we are so hoping for Fragile X families that we can really make a difference. And we wouldn't be doing this if we weren't dedicated to that. As a neurologist, seeing the patients, and we thank the FRAXA Foundation so much for having shown us families and patients so that we can understand what the disease is really like for families and for patients. And so as a neurologist, I just, this is something that I really, really, really want to work on.
Well, I can tell you as a mother of a young man with Fragile X, there's nothing more important. I may be a bit biased, but yes.
I totally agree.
There's nothing more important.
Exactly.
Well, thank you, Liz, and thank you to everybody who's joining us today. This is the first in a series of one-to-one interviews that we plan to do. So be sure to follow FRAXA on your favorite social media platform. We're on Facebook, Twitter, Instagram, LinkedIn, and of course, YouTube and the web. Thank you, everyone.
Thank you. Thanks, Katie. It was really fun talking to you.
You too.